Welcome to the Pediatric IBD Digest, the podcast focused on pediatric inflammatory bowel disease, Crohn's and colitis in children and young people.
We as your hosts, Dr. Tracy Coelho and Dr. Chris Bakewell, discuss clinical care and emerging research on select topics to support families and professionals alike. Please remember, the views shared are educational and do not constitute medical advice. Hey Chris, we are back with yet another episode on genomics. We have explored NOD2, dabbled in monogenic IBD, and today we are diving again into the deep, murky but fascinating waters of IBD genetics. Now, do you think our listeners may have reached their saturation point for genetic podcastomics?
Yeah, hi Tracy, it's really nice to see you. I was just thinking, it's been an absolute age since we recorded a podcast, so I suppose we should say welcome back to everyone. To be fair, it still amazes me that anyone listens in the first place, so we're extremely grateful for the support and the interest. And in answer to that question, I do think that genetics and IBD is one of those really complex topics that we could probably make an episode on once a week for a year and still not even scratch the surface. But today's topic is, I think, a bit more mainstream for the jobbing pediatric gastroenterologist. And I understand that we've got the perfect guest to guide us through, I guess, hence the Irish music you're playing.
Chris, you've got a very good ear. So yes, that's Irish folk music, and it's not by accident. I'm setting the stage for a special guest who hails from the Emerald Isle. She has a proud Irish background, and I must admit, I'll have to be extra careful with my words today.
Oh, okay. Well, now I'm interested. Tell me, why the trepidation?
Because our guest is a colossus in the field of IBD genomics. For a change, she is not a gastroenterologist or a clinician, but a leading academic whose brilliance is genuinely intimidating, in the best possible way. Now, she also happens to have been my PhD supervisor a few years ago, so you can see why I'll be tiptoeing on my linguistic diplomacy today.
So you must be talking about the one and only Professor Sarah Ennis, right? Now, from you and pretty much everyone else, I've heard that she's an absolute powerhouse in this field, and I presume you've got something extra special to welcome her.
Exactly right. I know Sarah is not a big fan of grandstanding, but she's truly a towering figure in the genomic space, and statistically speaking, somebody with a very, very significant p-value. So here we go. Aligned in impeccable brilliance to the academic sequence of the genomic arm, annotating and decoding DNA variants of profoundly real or seemingly unknown harms. Ladies and gentlemen, as the enhancer hums and the promoter strums amidst the fanfare of the transcriptional drums, please welcome Professor Sarah Ennis, a truly brilliant personality and not just a legend of genomic charms.
Welcome, Professor Sarah Ennis, to the podcast.
My goodness. Well, thank you very much for having me. That was a very unique introduction, not the sort of thing you expect at the normal conference. Absolutely. But yeah, delighted to be here.
Well, thanks for coming. I guess we'll jump right into introductions. And Sarah, I have to start by saying that your reputation precedes you, especially for me, having worked in Southampton with the Peds Gastro team for a while. You are Professor of Genomics at the University of Southampton, but I understand that you began your career in biochemistry. So my first question would be, what sparked your interest in genomics?
Right. Well, that goes back a very, very long way. I suppose I grew up in the west of Ireland. There wasn't very much to do. I spent a lot of time out in the countryside, and I used to, it's not legal now, but back then it was legal, to take tadpoles from the local pond and watch them grow. And I used to do that. I would take them and put them in a Jacob's biscuit tin with pond water and watch these tadpoles grow into frogs. And I would see them every day, and I would see this metamorphosis happening straight in front of my eyes. And I could not understand how that happened. I just couldn't, it just did not sit with me. I could not make out how the hell does that happen? How do you go from this wiggly little thing to a fully formed frog and they all come out, well formed and ribbiting and hopping about the place? So that was the very start of, I just couldn't sit. I couldn't figure out that puzzle. And then, of course, I was doing biology at secondary school, and I very much liked that. And when I went to university, I realized that a lot of the answers were held in biochemistry, trying to understand the molecular interactions. And of course, the recipe book for biochemistry is in the genome. So I think that's the pathway of how I ended up here. I was trying to get answers as how do you go from a tadpole to a frog? How do you go from a fertilized embryo to a healthy or an unhealthy human being? And I suppose the most valuable way I can see to apply that knowledge and the answers to those puzzles is trying to work out why are people sick and how can we get them healthier? And that's, I suppose, my reason for getting up in the morning and working in clinical genetics.
Charles Darwin would be proud.
Yeah. No finches, though.
Amazing. Thank you. Sarah, the Genetics of IBD study Southampton has been running for over 15 years. I know I have been part of it for a very long time. Possibly I was a little tadpole in your laboratory who hopefully has grown into a frog. But for our listeners, how did your research focus shift toward inflammatory bowel disease, and what were the key goals at the outset?
Yeah. So around the turn of the century, or soon after, in about 2003, the map of the human genome reference sequence became available. And that was a line in the sand that completely transformed how we look at genetic data in humans. We got to realize how frequent variations and changes in the genome were. And we realized that even common ones occurred about 3 common variants every 1,000 base pairs in the genome. And we've 3 billion base pairs in the human genome. So all of a sudden we had this abundance of data, and people started doing these studies called genome-wide association studies. And there was this thing called the Wellcome Trust Case Control Consortium, and it focused on a number of the most common diseases: type 2 diabetes, arthritis, and inflammatory bowel disease, with both subtypes, Crohn's disease and ulcerative colitis, were considered in there. And so I was very much involved in using these genetic markers to try and statistically track causal genetic signals across the genome using this abundance of genetic variant data. And so I was reading a lot around inflammatory bowel disease. In actual fact, we only knew about 1 gene involved in inflammatory bowel disease at the turn of the century, and that was NOD2. But all of a sudden, throughout these noughties, we actually got into over 200 different genes individually implicated by these genome-wide association studies. And so I was very much involved in developing and applying the methods to do that statistical dry lab analysis. And to be honest, at that point, I was relatively agnostic of the disease area that we were applying those tools to. But then just luckily, I ended up being given a student dissertation to mark around about 2007. And this student dissertation, it was a medical student who was working in the pediatric gastroenterology department. And I got to realize exactly how many children were being referred into University Hospital Southampton for a diagnosis of either subtype of inflammatory bowel disease. And I thought, goodness, there are a lot of children there. There's a huge referral. And of course, at that time, I was thinking that the strongest genetics are likely to be in the most severely affected individuals, but also the youngest diagnoses. So I expected the strongest genetic effects to be in children. And I could see that lots of children were being diagnosed in Southampton. I thought, well, here's an opportunity to move from, identifying there's 200 different disease genes implicated or associated in inflammatory bowel disease. But in actual fact, if you go into the clinic, if we went in and sat in your clinic or in Mark Beattie's clinic or anybody else's clinic in Southampton, not one little bit of those data were being used in the clinic. And I felt frustrated by that because I find the biology, the molecular biology, the genetics fascinating. fascinating, but I don't want it to be just academic. I want it to be useful. I want it to help make a personal diagnosis for patients and help get them on the better medications, or the medications that are going to make a difference for them. And so it was just this serendipitous coming together of emerging information in the area of inflammatory bowel disease, the fact that there were so many children coming through Southampton Hospital, and the fact that I wanted to cross that bridge between the academic identification of association and making it relevant to patients. And so that's why we started off and we got the ethics to start recruiting patients to the Southampton Genetics of IBD study. And we recruited our first patient just when I got back from maternity leave. I think it was October 2010. So yeah, we've been running almost exactly 15 years now.
Wow. Now, listeners at this point will be shocked, because this is the quickest we've ever got into anything scientific or interesting in relation to IBD. We're usually still on background, so I'm going to strike while the iron's hot. This is really good. I like this. So leading off of that, I guess it's a fair point to say that genomics and the field of genomics in IBD has transformed dramatically over the last 2 decades. In no small part due to your study. How does our understanding of IBD genomics, how has it evolved in that time? Could you give us a kind of state of play where we are now?
So I definitely think we're right smack bang in the middle of the process. We're nowhere near the end of it. So I do think there were some absolutely seminal papers in the last century, in the 1990s, identifying families with a preponderance of Crohn's disease or ulcerative colitis diagnosis. But of course, as I mentioned before, we really only knew about one single gene, and we didn't know then exactly how is that gene NOD2 predisposing to disease. There was some suggestion at the end of the last century that it was acting in a recessive form. But then that signal and that idea got obfuscated during the noughties, when everybody was just talking about single variant association tests and the fact that, oh, well, this individual has the high risk variant, but they don't have disease, therefore it's incomplete penetrance. Sorry, that's a lot of genetic jargon there, but it's suggesting that the relationship between the gene, dominant inheritance and recessive inheritance, got sort of lost and muddled during those association studies in the noughties. And then at the same time as that message about NOD2 was becoming a little bit less clear, it became obvious that lots of other genes were involved. So I think where we stand now is that we know there are about 7 or 8 different pathways, genetic molecular pathways, that are involved in driving disease. And so all the different 200 plus genes that have been implicated are involved in hypo or hyperinflammation, integrity of the epithelial lining, autophagy. So these key molecular pathways are involved. But what we are now also starting to do is really tease apart the clinical molecular detail that some individuals, they may present, they may look like barn door Crohn's disease, but in actual fact, sometimes they have a single molecular change or a single genetic change is really representing the majority of the driver in individual patients. And I suspect that number, it might not seem very much, but I suspect that maybe about 1% of patients diagnosed with inflammatory bowel disease actually don't have this complex autoimmune disease. They actually have just an immune problem caused by one gene. And by discovering that one gene, there very often is more clarity and more information that a clinician can take into account when prescribing the best clinical management for that patient. I suspect NOD2, and I think we've published on this, I actually suspect NOD2 accounts for a sizable proportion, 8 to 10% of Crohn's disease. And I think we are getting closer and closer to understanding that. So between NOD2 and then all these other very rare primary immunodeficiency monogenic IBD disorders, that you will definitely be up into maybe low double figures of pulling out individuals that their diagnosis will actually change from, for example, Crohn's disease to XIAP deficiency or CTLA4 deficiency or REL-A associated disease. And so it will actually be the genetic dysfunction or malfunction that defines the disease and that informs on the best and most appropriate clinical management. But I think we are very much in early days of identifying those monogenic disorders. And of course, once you move off the monogenic disorders, the next most likely thing is where two genes are interacting to cause disease. And it may be two genes in the same pathway. And I think we just bit by bit, we need to sort of start with the low-hanging fruit, the monogenic disorders, and move up the tree to the just slightly more complex. And then ultimately you will get to the very, very complex disorders, which are likely to be perhaps the maybe later onset and the most easily treated forms of disease, whereby you have interplay of maybe 8 or 10 different genes, all slightly perturbing the immune, systematic detection of microbes or the response to microbes, that they are actually, it's a complex interplay that's manifesting in disease in much later onset. But I do think genetics has a lot of promise to identify those monogenic diagnoses and those diagnoses where at least one or a small number of genes is the predominant driver of disease. And by attacking that or treating that central dysfunction, that you can bring about good health quicker.
Thanks, Sarah. That's really comprehensive. And now, you're obviously extremely intelligent and very knowledgeable about the genetics of IBD, but us pediatric gastroenterologists, clinicians, sometimes, myself included, we like to think of things more simplistically, and we're perhaps obsessed with this classification of Crohn's disease versus ulcerative colitis, IBD, putting patients in these clinical groups, which are not necessarily genetic groups. But I think a fair proportion of our listeners are likely to know that there are multiple genetic loci which overlap between Crohn's disease and ulcerative colitis. In terms of the underlying pathogenesis of IBD in general, do you think that kind of shared overlap, thinking outside of the boxes of the clinical groupings, tells us a bit about the overall pathogenesis? You know, have we got it right in the way we're classifying them, or is this a much more of a murky picture?
Right. So I suppose one of my hopes is that over time, we will begin to tease out these individuals with monogenic drivers of disease, and they will no longer fall under either of those or those three categories: Crohn's disease, ulcerative colitis, or IBDU. And I would hope actually that we do change from these. I view those very much as umbrella terms. We do find a genetic relationship, but I think HLA is much more likely to be a key player in ulcerative colitis. So that's the human leukocyte antigens and those immune genes all falling on chromosome 6. So I think HLA is much more likely to be involved in ulcerative colitis. And I think in Crohn's disease, you tend to have some of these more severe phenotypes that may end up having a higher representation of the monogenic disorders. But my hope ultimately is that eventually we do away with those terms and we actually define on the basis of the molecular drivers of disease. Increasingly in our work, we're trying to move away from using CD and UC and using much more refined and granular clinical phenotyping details on the patients to get a much more holistic and detailed clinical view of the patients that doesn't just pop them in a CD or a UC box. We actually want to know what are the presentations, what are the findings, whether it be stricturing disease, granulomatous disease. So what do we see on imaging? What do we see on histopathology? And precisely what anatomical regions is that related to? But to be honest, we're also looking beyond the gastrointestinal tract as well. So we're also getting those comorbidities, which patients also have psoriasis, which patients also have rheumatoid arthritis. Because for those individuals with multiple comorbidities in the autoimmune area, it's very highly likely that those individuals are much more likely to have a key driver that's impacting systemic autoimmune function rather than specific to the gastrointestinal tract. So I suppose the data I would like are not the UCCD badges. I want to know all the ICD-10 codes or all the SNOMED codes relating to that entire individual, whether it's about their skin, their lungs, their joints, or any other clinical manifestations. So it's that milieu of clinical presentations. That's what we want in research as our clinical data with which we integrate with genomic data.
Thanks, Sarah. As clinicians, I think we are still quite obsessed with those labels of CD, UC, and IBDU, but hopefully it will change over time. Now, if I circle back to the genetics of IBD study, which now includes both pediatric and adult IBD patients, as well as their family members. Now, I know to begin with, it was all pediatric patients, and then we went through an amendment. We had adult IBD patients included as well. Now, for our listeners, why might pediatric IBD provide specially valuable insights into the genetic basis of disease compared to adult IBD?
Yeah, that's a great question. So I suppose across all of genetics, the assumption is the earlier the age of onset or the earlier the diagnosis, there is this sort of blunt association between the age of diagnosis and the likelihood of a genetic cause. So if you have a baby born with a congenital malformation, that is a genetic, the cause there is a genetic disorder or a genetic cause. Whereas if you were to present with a slightly, some digestive tract issues in your 70s or 80s, that might be just overall sort of wear and tear on the immune system, perhaps a lifetime of exposures to different bugs and much more environmental component to much later onset disease. So the assumption was when we started this study and targeted on recruiting pediatric patients was that they would have the strongest genetic signals. And that's why we started recruiting in pediatrics. And I suppose I still expect that to be largely true, that where you have a really nasty perturbance to your immune function, you're unlikely to make it to 50 years old before being diagnosed. You're much more likely to be diagnosed at a much earlier age. And so that's why we started recruiting in children, because I wanted those strongest signals between disease and gene to be apparent. But then we did, as you say, about 2015, we decided to extend recruitment to be from the adult clinic. And that was for two reasons. One, we realized a lot of the adults were actually diagnosed as children, albeit they were currently being seen in the adult clinic. But also we realized that for some of these immune disorders, in actual fact, age of onset can be somewhat later. And I know for some of the monogenic diagnoses we're now starting to make in our Southampton IBD cohort, in actual fact, we're seeing them equally frequent among those diagnosed as adults as those diagnosed as children for now. I expect overall, 10 years from now, we will see that our hit rate of getting to the molecular causal diagnosis is likely to be higher in the pediatric onset population. But right now, we are making diagnoses, these monogenic diagnoses in the adult cohort as well. And of course, genetics isn't just about what's causing the disease. Sometimes you're looking at genetics to understand, as you know, Tracy, the reaction to drugs, whether somebody has an adverse drug reaction or not. And that is completely divorced of what's causing the disease. Your ability to process a drug in your liver is that's nothing to do with your inflammatory bowel disease diagnosis. And so if we're trying to understand, well, which individuals will respond or need a high dose or a low dose of drug, or maybe are likely to have a poor response to some drug, we are just as well just as capable of finding those answers in an adult cohort as we are in a pediatric cohort. And by having adult population, about half of our cohort right now have been recruited as adult patients and the other half as pediatrics. So we double our power, we double our sample size to observe these relationships between drug response and individuals, and looking at these pharmacogenomic signals. So those are not in any way age related. They are related to whether you've been prescribed the drug and exposed to this thing or not.
That's perfect. As always, kids are ahead of us. Even their genomes have more to say than us. So slightly different question. Now, you hold a substantive academic post at the University of Southampton as Professor of Genomics, and you collaborate with clinicians at the University Hospital Southampton nationwide and abroad across several specialties, not just gastroenterology. Now, how do you see the interface between research and clinical practice working in genomics, and what are the key challenges at that boundary?
Yeah. So I think the most successful clinical research is done at that direct interface between research and the clinic, mainly because the best clinical data are with the patient and with the place where the patient is being treated. So rolling back to those genome-wide association studies in the noughties, the sort of data that researchers were getting at that time were, does a patient have Crohn's disease, yes or no? It was literally a 1 and a 0. For Crohn's disease 1 and no Crohn's disease 0. The granularity of the clinical detail was quite honestly rubbish at that time. And now, as we are sequencing, as opposed to doing genotyping as we were during association studies, now we're actually sequencing large tracts of the genome and finding both the common and the rare variants in individuals. We have greater than ever need for the granular clinical detail, and you will only get that by working right up close beside the clinical systems, be it primary care, secondary or tertiary care. And some of my earliest meetings this morning were with those individuals who are coding that granular clinical detail in hospital systems. And we need, as researchers, to find safe and secure ways to access that granular detail so that we can continue to make improvements in making genetic diagnoses and predicting and prognosing and treating patients. So whether it's an inflammatory bowel disease or autoimmune disease or systemic lupus or Parkinson's disease, it's having that level of clinical detail is what will be essential to transforming and to unlocking our understanding of the genome, in my opinion. And so we're at a really exciting place right now, because HDR UK and the secure data environments that are being set up around England and the UK are facilitating the extraction of this high integrity coded clinical data. And I think there's things like large language models that can be used to read tens of thousands of records at the same time in a way that humans can't. Of course, there must always be humans in the loop. No piece of software is ever going to make a diagnosis, but a piece of software can read tens of thousands of records in a matter of hours in a way that a human can't. And so if we take careful baby steps, we can actually unlock this granular clinical detail, merge it and integrate it with the genomic data, and start to find patterns between the specific clinical manifestations and specific clinical pathways or genes within those pathways.
Sounds like a beautiful synergistic relationship. It's nice to know that you need us and we need you.
Absolutely.
Now we hear, of course, more and more, not just about genomics, but multi-omic approaches to IBD. And it's something that we talk about a bit on the podcast. But just, would you be able to explain to our listeners what we mean when we say multi-omics, and why would that be important when we talk about IBD research?
So, I don't know what was the first omic, but certainly genomic is the one that's my bread and butter. And that genomic is the study of all genes across our genome. We've got somewhere in the region of 25,000 genes. And genes are the recipes that code for proteins. And those proteins are the little machines that do the job, whether it's turning a tadpole into a frog or making your immune system work well to recognize a bacterial pathogen in your gut as non-self and nasty bad bacteria against which you need to mount an immune response. So that's the genome, and that's entirely encoded in the DNA in the nucleus of all your cells. But the bits of the genome that are made to go to work are very different in an eye cell to a liver cell to a gut epithelial cell. And you're not using all recipes, all of those 25,000 genes at the same times in all these different cells. You're more than likely just using a subset. So as genes are being used, we transcribe. So we take a copy of the gene that's in the nucleus, and we take a copy of it and turn it into RNA, messenger RNA. We take that outside the nucleus, and we use this cytosolic machinery to turn that messenger RNA into a protein. And so some people, and these different omics disciplines used to be quite siloed. Geneticists or genomics, genomic informaticians would work in genomics, and other people would work in transcriptomics. And transcriptomics is that copy of the gene that's been taken from the copy is taken in the nucleus and taken out into the cytosol. That's the transcript. And so transcriptomics is actually sequencing what is being used in any given cell at a given time. So rather than looking at, genes that are involved in neuronal signaling, we don't need to necessarily look at that in a gut epithelial cell, but we might want to look at, well, how much is NOD2, for example, being expressed in the ileum? How much is IL-10 being expressed in the ileum? And you can actually do sequencing of the RNA transcripts to inform you as to which of these recipes are actually being functionally used at any given time, or importantly, not functionally used when you expect them to. And so transcriptomics is the readout of those messages of which genes are being used. And of course, then proteomics is when those messages have been successfully translated into proteins. So they're different readouts. With, I suppose, at the base level, you have the genome. And then if you go into specific tissues or cells, you see, well, which bits of that genome are being functional, and that's the transcriptomic, and you're actually sequencing RNA there, not DNA. And then proteomics, you could use chemistry such as Olink to detect the proteins that are most or least abundant in any given cell at any given time. So those are the three omics that are probably most often used, but you also have metagenomics, metabolomics. There's many, many other omics. I suppose metagenomics is an interesting one from an inflammatory bowel disease perspective, in that that's getting a sample, for example, from a gut biopsy, but you're not actually sequencing the human there. You're sequencing all of the bugs, and you're identifying what bacteria, what's the set of bacterial species that are identified in the gut. Or viromics is when you're identifying all the viruses that are identified in any sample or any tissue at any given time. So you seem to be able to add omics onto almost anything these days, but I think genomics and transcriptomics and proteomics are the three that are most often layered to try and help explain one another these days.
Sarah, just to let you know that I and Chris have developed a new science, and it's called podcastomics.
Well done.
Yeah, there's not a lot of evidence behind it, I will say. Yeah, perfect. Obviously, and what that speaks to is the layers of complexity. That once you start to research this, I'm sure that you realize that what you thought you knew at one point then relies on knowing a huge amount of other information to put it into a clinical picture. And I guess that's not specific to IBD, as genomics becomes part of everyday NHS practice. Clinicians who are listening, I'm sure, will understand the headache of variants of unknown significance, or VUS. You send off these genetic tests, you get back a result that you have no idea how to interpret. Why is that challenging? Because, of course, that's going to come up more and more as people like me send off genetic tests.
Yeah, I can promise that the headache that is variants of uncertain significance is not limited to the clinic. Researchers are banging their heads against the wall very regularly about these. So it basically means a variant of uncertain significance is often, there's some variants that we can say, oh yeah, that's definitely going to kibosh the protein. It's a loss of function variant. It introduces a stop change, or it's a frame shift, which means that the when you transcribe the gene in the nucleus, the mRNA is incomplete and will get degraded by the cells. You just don't end up with the protein. It's very easy to interpret that loss of function mutations are going to abrogate the function of that gene and the protein product. What's much, much harder, and what VUSs are enriched for, are missense variants. So these are variants whereby you've transcribed the DNA into RNA, and the RNA has been converted into this string of amino acids that form a three-dimensional shape that is the functional protein. But that string of amino acids is made up of the 20 amino acids that's common to us all: alanine, leucine, isoleucine, tryptophan, tyrosine, all of these amino acids. But very often, variants of uncertain significance, they change one of those amino acids for a different amino acid. And it can be very hard to say, well, so what? If you change an amino acid, if you have a hydrophobic binding cleft of some really important receptor, and you replace one of the hydrophobic amino acids for a hydrophilic amino acid, well, then all of a sudden that receptor is going to repel its ligand as opposed to bind its ligand. So that's going to be absolutely catastrophic. So you might then say, well, is it an amino acid change from hydrophilic to hydrophobic? Is it at a binding cleft? And you try and infer whether or not that variant might cause change. But the problem is, when we sequence humans, patients, participants of big research studies, we very, very, very regularly uncover variants that we have never seen before. So missense changes that nobody has done a biochemical test on. And you might guess that it's in a binding cleft, so it could be important. But guessing is not good enough for clinical action. You must be certain. And therefore, that variant will remain a variant of uncertain significance until robust functional testing has been conducted, peer-reviewed, and published. And the evidence is there that that truly is a causal variant, that impacts the function of that protein and therefore is likely to be the underlying cause of that disease. And so there is this threshold of evidence that must be met before you could say that missense variant in that protein is likely to be the causal diagnosis for this patient. And if that threshold is not met, the variant will remain a variant of uncertain significance. And it's an area of huge frustration for both researchers and for clinicians. But we must hold the bar very high. You cannot be making life-changing decisions or incredibly expensive, stem cell transplant, bone marrow transplants, huge cost drugs. You cannot be making those decisions on a guess, or even a strong guess. It needs to be a certain level of evidence that you have for it to be a variant of certain significance, for you to make very important clinical decisions. There was at one stage, there was a grading of variants of other uncertain significance, and they were all by whether it was kind of smoking hot to where it was quite cool. And I think those variants that we really, really think, it's in a region that has been previously identified to be enriched for variants that are nasty and pathogenic, then individuals and researchers will prioritize those variants for functional testing in order to be able to move it from a VUS to a known pathogenic variant. But the problem is we observe very, very rare things incredibly frequently. And the rate of that functional testing lags way behind our discovery of these variants. And that's the bottleneck. And it's where things like AlphaFold are coming now. So they're trying to use AI to draw 3D or to map 3D structures of proteins with and without the missense. But it's still in silico inference. It's it's adding levels of evidence, but it's not necessarily getting us over that finish line to be able to say on a clinical diagnostic report, this is the causal variant. So it is an area of frustration. There are multiple efforts around the world in trying to speed up the interpretation of these VUSs, but I'm not going to lie, we're not going to get an answer for that anytime soon. We are still uncovering them at a far greater rate than we are resolving them.
That's perfect. I think you have addressed this question with a very high level of genomic eloquence. Now, as we have just discussed, with genomic testing becoming part of point-of-care diagnostics, clinicians are increasingly faced with the challenge of interpreting and explaining complex genomic reports to patients. And I think clinicians today need a stronger grounding in genomics than ever before. So from your perspective as an educator, how well has the medical curriculum adapted, and what more still needs to be done?
Yeah, I think there's been lots done. NHS England funded a Masters in Genomic Medicine. We ran one of those here at the University of Southampton from when they were originally funded by NHS England up to today. And that was a master's degree course for anybody working in the area of medical genomics. But of course, on the ground, you've got lots of people who have already qualified, already specialised, and all of a sudden they're having to become cognizant and aware and able to interpret these genomic diagnostic reports that might be coming through. And I think, like it or not, genomics are going to become even more mainstream than they already are. It's estimated that, in about 10 plus years, about half of all medical interactions will involve genomic data informing some aspect, whether it's the diagnosis or the level of the drug you should be giving or prescribing, or a metagenomic result to understand which bugs are there so you can target treatments towards the specific bugs. I think genomics are going to be increasingly common for all specialities, and primary care all the way through secondary and tertiary care. So it's not just at the master's level, it needs to be ingrained in undergraduate medical education. And I think that is becoming more and more au fait. I know that in the teaching I do to first year undergraduate medicine here in Southampton, I link biochemistry, metabolic biochemistry to genomics all the time. And of course, the genetics and genomics as a subject, that has evolved substantially over the last decade. And students are learning more and more about the National Genomic Test Directory and about the role of genetics in whatever their area of clinical expertise. So I think we've made a very good start. But I think there's still probably even more work to do. And it's probably a legacy of the fact that many of the people who are in the current system probably learned less about genomics when they were doing their undergraduate degrees than is probably required these days.
So more of genomics and less of the Krebs cycle.
I love Krebs cycle. Krebs cycle, there are very many mutations that can be involved in metabolic biochemistry.
I'm sure you still have the big picture of the Krebs cycle in your office.
I do. Yes, I do.
Don't ask me to say anything about the Krebs cycle. I think, Sarah, you've done a really good job there. I mean, given what we know and given what you've learned and your study has shown about the genetics of IBD, where it's moving to in the literature is talking about this concept of precision medicine, i.e. preempting the right treatment for the right patient at the right time. How close, and I think I know the answer, how close do you think we are to that goal? And do you think things are progressing at a pace that you'd be happy with or you'd hope for?
I think for some patients, we have reached that goal. So for, it's around about 25% of ProBan's patients who have been referred to the Genomics England project. They have a molecular diagnosis, which revises their diagnosis and informs on their medical treatment. So we already have reached that precision medicine for a very, very small proportion of the population. What's going to change is the proportion of the population that we manage to make that step change with. So, for example, in the NHS right now, very, very few patients are eligible for genomic sequencing if they have a diagnosis of inflammatory bowel disease. They have to be aged very, very young, and they have to have a constellation of symptoms. Even if they're as old as 6, they have to have extra special lists of clinical presentations. Whereas what we're observing in our research study is actually somebody diagnosed in their 30s have a single monogenic cause of their disease. And that's what we're hoping to do by get the message out that actually if you sequence adult patients or patients who are not aged less than 2, there is still valuable diagnostic information. That precise diagnosis can be made in those individuals, and that there is a health economic benefit of employing sequencing in those individuals that will mean that you prescribe the correct treatment, get them back to school or work as soon as possible, and those individuals have less demand on the healthcare system and contribute to the NHS or the UK economy more effectively. So there will be, whether it's quality of life or NHS pounds, there will be benefit to everybody by implementing this. But we need to generate the evidence for that health economic argument first, and that's what our study is trying to do by applying genomics to a cohort of patients that are not actually able to avail of genomic sequencing in the NHS as yet. We're hoping to generate the evidence of value of doing that. And you don't need to make a precision diagnosis for everybody to tilt that health economic balance towards implementation, more widespread implementation of genetics and genomics. So it's a very long-winded answer, but I think we have reached precision medicine for a tiny proportion, and what's going to change is the proportion of individuals we can apply that to. But it's as I mentioned earlier, it's not just for diagnosing, it will also be for prescribing. Sometimes it might decide how much painkiller you give or don't give, how much antidepressant you give or don't give, or warfarin. So it can be withholding drugs where the individual can have an inappropriate response, or modulating the level of drug that's given. So I think pharmacogenomics will be brought in there as well.
Sarah, I think we are at the final quartile of our recording. So your department is increasingly engaging with AI and large language models, which is very exciting. And when I attend some of the research meetings, the discussions can sometimes feel quite overwhelming in a good way. So how are these technologies beginning to impact genomic research and data interpretation?
So there's lots of different ways. AI is a huge category, just like medicine is a big category. Machine learning is one of the approaches that we might be using to find patterns in data. But I suppose one of the other ways that I hadn't anticipated going back some while of using AI is these large language models. It's unlocking, there are petabytes and petabytes of really valuable clinical information that are locked away in unstructured data in hospital systems, in any other NHS healthcare setting. And it is just not possible to get one person to sit down and read all of those things and code them into a way that can be used by research. That's not possible. Even doing that for, a handful of people would take one individual a very long amount of time. And of course, as soon as it's done, it's out of date, because what if they get ill tomorrow or having some other new manifestation tomorrow? So it's out of date as soon as you've done it. So it's just not something that humans can do efficiently or effectively. So large language models we're using in a way that's really transforming the quality of the clinical data we get to match up with the genomic data in reading radiology reports, imaging reports, pathology reports, and extracting from those reports the granular detail of what's happening in patient A, different to patient B, different to patient C. That's the biggest place where I find AI is making a real change to our ability to discriminate and better characterize the clinical presentation and the clinical journey of individual patients, such that we can map that pattern and that clinical manifestation against specific genetic changes that may be driving them.
So I think you have decoded quite a bit today. Thank you for such a fascinating conversation and for taking time to share your thoughts. But before we hit pause, let's just switch from serious IBDomics to something lighter, and we call it, I call it the gut fire round. And for this segment of the podcast, my co-host, Dr. Chris Bakewell, generally begins to emotionally disconnect and enter a state of podcast hibernation. But I'm still here.
I abstain.
So, Sarah, I've got a question for you. You have been invited to a dinner of a lifetime by 3 legendary names from very different worlds, dead or alive, ranging from a saint to a pop singer to a scientist. And I will give you 3 options. You got to tell me who is going to be at your table. So option 1, you have Ronan Keating, dinner at a restaurant of your choice. He will be asking you to rewrite one of his hit songs, When You Say Nothing at All, and he wants you to make it into a genomic hit, maybe something like When the Gene Says Nothing at All. And you will have to join him for a karaoke afterwards. So that's option 1.
Oh my God.
Option 2 is Saint Bridget of Kildare. She promises the best Irish food ever at the Kildare Monastery, so good that you'll never crave for another meal again. No alcohol at the monastery, but there's a possibility that the water will miraculously turn into Guinness. And if you spend a full day in prayer at the monastery, she will probably hand you the key to all the unknowns in IBD genetics. You have option 3, Gregor Mendel, a quiet dinner in the garden over a pea-based feast made entirely of genetically modified peas. You get to advise him on genomics and maybe gently explain that there's a bit more to inheritance than the green versus yellow. So Sarah, who would you choose?
Oh gosh, that is a very bizarre and eclectic options there. Fair dues to Ronan Keating. I'm going to set him aside. Karaoke is not my thing. Thank you very much. You had put me off St. Bridget because there was no alcohol in the monastery. But if we have a promise of an Irish pint of Guinness, I'm not too bothered about the Irish cuisine. Although the meat is great, but the actual recipes are not fabulous. But they've got great produce. But the bit that interested me about St. Bridget was at the end she was going to unlock all the mysteries of genomics. So I think if the Guinness is on the table and she's going to unlock everything, I think I'll head to Kildare and meet St. Bridget. Gregor Mendel, I have a huge amount of respect for, and I would never want to re-educate him.
So I'm sure the monastery will be overjoyed and singing in a chorus of Alleluia.
Tracy's gut fire round tells the listeners more about Tracy than it does about I guess.
I think so.
Quite possibly. But thank you, Sarah. That was a brilliant discussion. Thank you very much.
Oh, thanks for having me.
Yeah, thank you so much. That was really, really lovely.
So after chatting with Professor Sarah Ennis, I thought I would sneak up on one of her brilliant PhD students. And that's when I found Fernando, surrounded by monitors that looked like NASA mission control, all filled with alien codes. Fernando, welcome to the podcast. Hope these next few minutes feel like a mini vacation from the genomic tangles.
Hello, thank you for inviting me.
Welcome, Fernando. So, could you please give us a summary of who you are and what do you do and what are your interests?
Yes, so I am a third-year PhD student working on the genetics of inflammatory bowel disease. The main focus is on integrating genomic data with clinical data and the application of machine learning methods. I have a master's degree in bioimmunology and microbiology from the University of York, and some of my work revolves around characterizing disease burden of genes in key evidence pathways and looking for drivers of disease complications. In my spare time, I like listening to podcasts, such as yours and reading about history and science.
Oh, you love listening to this podcast. Thank you. We are blushing already. Fernando, your PhD integrating clinical and genomic data with machine learning sounds like absolute magic. I would have loved to ask you a million questions, but in the interest of time, let's jump straight into the gut fire round. You are going on a little voyage of imagination. You are a Spanish explorer and a genomic pioneer. The Royal Highness, the King of Spain, himself has commissioned you to set sail across the vast genomic ocean in search of a mysterious island which is said to hold a treasure chest containing the ultimate secrets of human genomics. Now, you will be leading this powerful genomic fleet with your genomic binoculars, joined by world experts, including Professor Sarah Ennis, racing against other nations to reach the island first. So, tell us, I've got 4 options for you. What would you name your fleet? So, option 1, the Genomic Conquistador. Number 2, the Genomic Armada. Number 3, La Flota Genomica del Tesoro. Now, please excuse my atrocious Spanish. And option number 4, the Genomic Columbus. Now, or would you rename it entirely and make a mark in history with your own name?
I think I'll go with option 2, that's the Genomic Armada.
And why would that be?
Well, I guess it has a nice ring to it. Genomic Armada sailing across the ocean.
Okay, all right.
Yeah, there's a lot of historical background that I think you have to ignore when choosing one of those names. But
pure coincidence, I promise. That's amazing, Fernando. With your genomic fleet, I'm sure you will sail straight to the genomic treasure and rocket through your PhD in full flying colours. Thank you for joining us.
all the best with your research.
Yeah, thank you very much.
Dear listeners, thank you for tuning into this episode of High Dose Genomic Shenanigans. Hope you enjoyed. Hope to see you soon. Bye-bye.